Hey everyone, it's Chow here, and today we are going to briefly go over an overview of the immune system, so let's get started. Now, we obviously can't talk about the immune system without briefly discussing the lymphatic system, which actually allows for circulation of fluids within our body. Now, it's actually separate from our circulatory system, but is associated with it. So, interestingly enough, whenever you eat lipids, so fats, your body actually breaks down the fats and then passes those fats in the form of chylomicrons across your lymphatic system before it actually drains into your blood. So it's separate, but there's also sort of a linkage between the two systems. Of course, with the lymphatic system, we're talking about that movement, right, of lymphocytes between the blood and the lymph. And we have some interesting lymph tissues such as bone, the thymus, as well as the lymph nodes and the spleen. Of course, that's kind of more or less trivial information in regards to the lymphatic system. What we're really interested in is how does the body respond to foreign invaders? So if, for instance, a virus from the outside world comes into your body, there are three phases the body has to go through to express some kind of an immune response. The first step is for recognition. So obviously, if a virus comes into the body has to recognize it. More importantly, it has to recognize that virus as foreign and potentially dangerous and not think of it as part of the body. So recognition is the first step and is quite critical. Next is activation. So that's when the body actually goes and mobilizes certain cells, for instance, maybe B cells, to proliferate and ultimately fight off the invader. So that's activation. So you're mobilizing whatever cells we need in the body, whether it's B cells, whether it's T cells, etc. And then finally, we have the effector phase, which is when those cells and molecules actually go out and destroy the invader. So this is actually sending your troops into battle to fight off whatever foreign invader, like a virus, that's in your body. So the three phases of the immune system, pretty straightforward, I think. Okay. So there are some cells of the immune system. The key group of cells are the leukocytes, which are your white blood cells, and they're broken down into two main categories. You have the lymphocytes, which include your B cells and your T cells, as well as your phagocytes, which basically include your macrophages, dendritic cells, and they are the ones that ultimately engulf pathogens. Your T cells are sometimes going to help destroy certain infected cells. Your B cells are going to be the ones that are producing antibodies. So speaking of antibodies, which is what we often think about when we talk about the immune system, here's an antibody, also known as an immunoglobulin. So it's actually a tetramer that's linked by disulfide bonds, and there are kind of two main regions. We have a constant region and a variable region. The variable region is going to be the one that's specific to one type of antigen, whereas the constant region is going to help designate function. So interestingly enough, there are actually 5 different classes, and that allows for variation in the function. But ultimately, the constant region is the one that stays relatively similar between the same set, whereas with the variable region, we see variation that are more of an antigen specific. So there you go, antibodies. Now, as I mentioned, antibodies are going to be produced by B cells to allow for some kind of effect, hopefully to sequester whatever dangerous chemical or bacteria or virus that's found within the body. And we can actually use these antibodies to tag certain molecules. So if we have a part of a virus that's in the body, we can use these antibodies and bind to them and tag them. The tagging ultimately can potentially just sequester and prevent the virus from being active. So the simple act of the antibody binding to the virus might already be enough to stop it. More likely than not, however, the binding of the antibody will allow for the sort of signaling of nearby cells to come in and help with the process. So it can actually call in, the binding of the antibody can basically light up and act as a flag to draw in, for instance, macrophages to come in and basically destroy the virus. So it basically neutralizes and ultimately allows for that particular dangerous virus or particle to be destroyed. Now, what's really fascinating is how in the world do we have such a great diversity of antibodies? This is partially helped by this process known as a rearrangement, more specifically of a big old supergene. So in order to keep things straightforward, I will just mention that it happens at the DNA level. So you have splicing and cutting of certain portions of the genome, and this variation, right, within successive rearrangements allow for immunoglobulins to be extremely diverse. And this rearrangement is what ultimately causes variation within antibodies, as well as also T-cell receptors, which are quite important for the system. Okay, so what happens when you actually have some kind of a cell that needs recognition? What happens if the virus, for instance, gets within the body of a cell and isn't floating around? So obviously if it's floating around, you can just have that antigen and then the antibody might be able to just bind to it directly. But what happens if you have a virus that entered into the cell? Well, there are ways to actually help allow for an immune response with that as well. And this is with this over here, the major histocompatibility complex, also known as the MHC. So these are just proteins that are found on the surface of the cells that can help show and present proteins of within the cell itself to the outside world, which allows for T helper cells to come in, bind to it, and recognize and see whether those proteins are actually part of the body naturally or something foreign. So there are two MHC systems. We have MHC1 and MHC2. MHC1 is found on almost all cells, and they actually allow T cells to recognize certain infected cells. And so you have just a cell that's going to be posting out some kind of a peptide over here, for instance, and then the T cell might come in, the T helper cell might come in, or a cytotoxic T cell might come in, recognize that, and fire off some toxins to destroy that cell if it's something an infected or problematic cell. You also have MHC2 systems, which are slightly different because MHC2 systems are only found on antigen-presenting cells. So antigen-presenting cells include things such as B cells, as well as macrophages. Dendritic cells, I believe, also have them. And what's really fascinating is that the MHC system allows for presentation of non-self antigens. So you have a macrophage that engulfs maybe a virus or a bacteria, and it presents the proteins of that virus or bacteria to the surface, and that allows for T helper cells to come in, bind to it, recognize it as something bad, and then signal, for instance, for B cells to make some antibodies or some kind of a memory B cell in order to help mediate a humoral response in this case, whereas MHC1 often targets for more of a cell-mediated response. We're going to talk a little bit more about this in probably the adaptive immunity video, so that's something for the future. Okay. Now, we also have these things called T-cell receptors. So the T-cell receptor is found on different types of T cells, and it's an integral membrane protein that allows for recognition of the MHC antigen complex on other cells. So in this case, we have MHC class 1, so it's just a normal cell that's presenting via an MHC class 1 method, and it's going to be recognized by a CD8 receptor. And then over here we have MHC class 2, and that's going to be on an antigen-presenting cell or an APC cell, such as a dendritic cell or a macrophage or a B cell, and that's going to be recognized by CD4. So different variation in recognition, but ultimately the main goal still is to allow for defense of the body. So what happens after you have something like this that happens? So imagine you have an antigen-presenting cell. It's putting out some kind of a really nasty protein from a very bad virus. The T helper cell recognizes it. What is it going to do? Well, most likely what it's going to do is actually release cytokines. So there are many different types of cytokines, and these are soluble cell signaling proteins that ultimately bind from one. It's going to be released from one cell to the surroundings, and it's going to bind surface receptors and ultimately alter other cells' behaviors. So it's going to be very important because the cytokines, which in a way are kind of like hormones, but they're not completely like hormones, and there's going to be a big debate as to whether these are truly hormones or not. But these cytokines are going to be released by cells to help signal other cells about potentially needing help. So it's going to potentially help stimulate the movement of other cells towards sites of infection, towards sites of inflammation, and maybe even sites of trauma. And that ultimately allows for that signaling and connection and communication between different cells, and hopefully that will allow for some kind of a positive response to allow your body to protect itself against whatever issue is occurring, for instance, a viral infection. So there you go. That is a brief ramble on the immune system. We will actually talk a little bit more about adaptive immunity as well as innate immunity in the next couple of lectures, so stay tuned for those. But as always, I hope you all found this mildly useful, and best of luck studying, and I will see you in the next video.